Is Expasy, Expert Protein Analysis System. We need to know what it is. Expasy is the SIB bioinformatic resource portal which provides access to scientific databases and software tools in different areas of life sciences including proteomics, genomics, phylogeny, systems biology, population genetics and transcriptomics. What is its background? It was created in August 1993. It was one of the first web servers for biological sciences. It has undergone constant modifications and improvements. In June 2011, the SIB Expasy Bioinformatics portal was launched by the SIB Swiss Institute of Bioinformatics. Expasy has been designed, developed and maintained by the SIB web team in cooperation with several other SIB groups and Expasy user. Now we will move to the Expasy. Select this link. So Expasy server is dedicated to the analysis of protein and protein structures. Sequence analysis tools are different that include DNA to protein translation, pattern and profile searches, post-translation modifications, primary structure analysis, structure prediction, 3D structure predictions, alignment and many more. Now we will discuss the visual guidance in which there are 6 elements. Select the DNA. Here we have the keywords sequence alignment and sequence analysis. DNA is used for the sequence alignment and sequence analysis, and these are the tools that are used for this. And now RNA, these are tools for RNA that are used for next generation sequencing and for transcription module. For the proteins, now we see the organism. So these are the tools for the organism which check the transcriptional factor, binding site, transcription molecule, regulating transcription, start site annotations, and biochemical pathway also. Pathway tools used for the next generation sequencing pathway. And now we will discuss the categories in which we will further discuss the proteomics that explain protein sequences, identification, protein experiment, functional analysis, sequence size, features and motifs, protein modification, protein structure, protein interaction, similarity search and alignment. And for this purpose there are many tools and databases used. There are many tools if we see, but here we will discuss only those tools that are commonly used. First of all, we will discuss the Swiss model workspace. Swiss models workspace is a fully automated protein structure homology modeling server, and it is providing 2 ways of doing homology modeling. First approach mode is fully automated procedure, and second is optimize mode, that is manually optimized sequence alignment via deep view program, which provide manual control during homology modeling and tools for analyzing, generating models. We can use this model by, first we have to create account in this, where we have to enter our email ID and then set our password. After login this site, we have to paste or target sequence of a protein and we enter the project title and the email ID, and they will send our 3D protein structure to our email ID. And this modeling Swiss model homologic modeling is performed to search the suitable templates and check sequence identity with targets. And if we discuss that what is the purpose of Swiss models workspace. So it provides the 3D structure of protein of or target protein that we can observe the structure 3D model of a protein. And it will compare the sequences of protein that are already reported, then it gives our 3D structure. And now we will discuss the Swiss model repository that has all protein structures, all protein structure that has been already reported. And we will have the enzyme database which have all the information of enzymes, and it explains the enzyme nomenclature. If we talk about the tools, there are several tools that we know. BLAST, where we paste or FASTA format, and then run the BLAST. Run the BLAST, and it will give the similar sequences of or proteins from different species. And then we will discuss the cluster W. It is used for the multiple alignment sequence, and basically it is used to do phylogenetic analysis, same protein from different species, and it shows the evolutionary relationship. And we will cluster W. Firstly, when we give our sequences and run the cluster W, if sequence have the similarities, then it will show steric sign and reveal the conserved regions in our sequences. And now we discuss embossed translation tool, where we have nucleotide sequence translation and protein sequence back translation. Here we will paste or DNA or RNA sequence, and then the next step, and here we paste our RNA sequence first, then second step is to select our standard code, and our third step is to enter email ID and enter title, then submit it, and we will get our protein sequence on our email ID. And now we will discuss the protein sequence back translation tool that is used for the reverse translation to convert protein sequence to nucleotide sequences. And here we paste our protein sequence. Second step is to select species, and we have used ubiquin and lactobacillus, that is from bacillus species. And third step is same as we discussed before. Enter we enter our email ID here, and then title, and then submit it, and after then we get our nucleotide sequence on our email ID. Next we discuss the translation tool. Expasy translation tool, which is used to translate translation tool, which is used to translate our RNA sequence or DNA sequence into protein sequence, and here we paste our RNA sequence, and next is click translate button, and here we get our protein sequence. This is our frame of reference 1, 2, 3, and I'm sure that our protein is start from methionine, and this frame of reference has 2 methionine and the sequence. And now we will discuss the other tools. As we all know, project tool is used for the stereochemical quality of a protein structure, and we use the check tool for the Ramachandran plot of protein structure, where it gives the similarity within the protein structure. And there are many tools, but we discuss only a few tools. Our mainly focus is to have a quick overview of the Expasy server. So we have discussed some important and commonly used tools. Thank you.